Hello everyone. In this video, we'll be discussing about the subcellular organelle Golgi complex. Right. Golgi complex is otherwise they known as dictyosomes. Right. So, what is the role of Golgi complex inside the cell? Okay. So, Golgi complex mainly involved in modification and sorting of proteins. So, as I mentioned earlier, right. So, this endoplasmic reticulum and Golgi complex, they are in close association with the nucleus. Right. So, whatever the protein synthesized in the endoplasmic reticulum, they need to be undergo modification. So in case of endoplasmic reticulum, they have one particular reaction in modification of proteins, that is hydroxylation reaction. That means addition of hydroxyl groups to a particular protein to make it active. Same way, Golgi complex, they also have some reactions which involved in activating the proteins. So such reaction is glycosylation of proteins. Glycosylation of proteins is nothing but addition of carbohydrate. Okay, addition of carbohydrate. There are some proteins which require carbohydrate to their function, right? So that reaction taken place in Golgi complex, and it is a major site for new membrane synthesis and its formation of lysosomes and peroxisomes. So this Golgi complex cistern are of three types: cis, medial and trans. Glycoproteins are generally transported from endoplasmic reticulum to cis Golgi, okay, and to medial and then to trans Golgi. Trans Golgi is maximum glycoprotein content. Okay, the trans membrane of Golgi complex has got maximum glycosylated proteins. And to say the finished products may have the following destinations. That means one out of the expected. They may pass through plasma membrane to surrounding medium. This forms continuous secretion. Example, secretion of immunoglobulins by plasma cells. Okay, so immunoglobulins are also protein in nature. Immunoglobulins are nothing but antibodies, IgA, IgG, IgM, IgE, right? So they are also protein in nature, and they are also supposed to be synthesized through protein synthesis, right? And second point, they reach plasma membrane and form integral part of it but not secreted. Okay, so when there is a need arises, then only they will be secreted. Otherwise, they are just packed inside. They are formed into secretory vesicle where these products are stored for longer time. Under appropriate stimuli, okay, the contents are secreted. So release of trypsinogen by pancreatic acinar cell and release of insulin by beta cells of Langerhans are cited as examples. That means what we are mentioning here, trypsinogen. So this is a proteolytic enzyme. Okay, proteolytic enzyme which is releasing from the pancreas. Same way insulin which is also releasing from pancreas. The synthesized materials may be collected into lysosomal packets. Okay, there will be, like as I mentioned, until unless there is a stimuli, there is a need, these secretory vesicles will not be secreting the synthesized products. So coming to the next one, lysosomes. The lysosomes are nothing but, what to say, they are vesicular structures and their size is like teeny, to say 0.4 micrometers. So they are surrounded by a lipoprotein membrane. So lipoprotein is nothing but combination of lipid and protein. And what is the main specification of this lysosome? They carry acidic pH. So rest of the cell having a normal pH around 6.8 to 7.2. But here the specificity of the lysosome is it is having acidic pH, that is 5. And the main role of lysosome in the cell is cleavage activity. Right, as I mentioned earlier, lysosomes are like scavenging activity they do have inside. That means any waste products or any pathogens or like any degraded, any lifespan completed substances are there, it will engulf, it will take them inside and they will degrade. Okay, and this is possible because of the enzymes, that means hydrolytic enzymes. That means they are group of enzymes which hydrolyze any type of particular substance. Okay, and the marker enzyme for lysosome is acid phosphatase. Acid phosphatase is enzyme which you can give example for hydrolytic enzyme. Okay, if you see the structure here, single wall membrane, this is lipid bilayer, you can say lipid bilayer as like a cell membrane, and enzyme complex, that means mainly acid phosphatase. So lysosomes are the cell garbage disposal system, as I mentioned. So lysosomes recycle materials by breaking down worn out parts of cell into smaller units. They deliver these materials to cytoplasm for using the constructing new proteins, right? So suppose I say hemoglobin. Hemoglobin is a part of like heme and protein part globin. So once RBC completes its life cycle, like 120 days, so RBC will be lysed, the main content of RBC is hemoglobin, it will come out, and this hemoglobin will be separated into heme and globin. Heme will be converted to bilirubin and transported to liver for detoxification or conjugation reactions, okay, make it to soluble. And globin, they are proteins, right? So they are taken by these lysosomes and they make it into individual amino acids, and these amino acids will be transported to a pool called amino acid pool in the body, and from there, whenever there is a need of these amino acids to synthesize particular protein, there they will be transported. So this is totally dependent on the activity of lysosomes. So they deliver these materials to the cytoplasm for use in constructing new proteins. At the same time, if the membrane of lysosome breaks, the enzymes released may also destroy the cell itself, giving lysosome the name suicidal bag. So remember, lysosomes. So in MCQ's question, they will ask which subcellular organelle acting as a suicidal bag. So lysosomes. Why? The membrane of lysosome breaks means the enzymes released, okay, the enzymes which are present in lysosome is acid phosphatase. It will not show any difference. So it will consider other subcellular organelles as a dead particles and it start engulfing and cleavaging them. So that's why it is very, very important to keep these acid phosphatase and other lysosomal enzymes in packed state or inside the lysosomes. Better not to release it out. Okay, so that is the main thing about lysosomes. And also lysosomes which are also required for intercellular digestion of biopolymers like cellular digestion, like hydrolysis of carbohydrates, proteins, okay, and nucleic acids. So all these, that means to break down high carbohydrates. What is the main linkage present in carbohydrates? Glucosidases, glycosidic linkage. For proteins like cathepsins, collagenase, elastase, lipids, fatty acid esterase, phospholipids. So they are all working on the linkage, carbohydrates, glycosidic linkage. So glucosidase acting on the glycosidic linkage. For proteins, these cathepsin, collagenases, elastases, they work on particular protein, like collagen is a protein, elastin is a protein, cathepsin also is a protein. So they work on the particular type of protein. Okay. And lipids, fatty acid esterase, the ester linkage present in lipids. So they break that ester linkage. And nucleic acid, the main linkage is phosphodiester bond. Okay. And they will break all the phosphodiester linkage. So clinical aspect. So there are like lysosomal storage disorders, right? Like inclusion of cell disease, like a protein target defect, accumulation of undegraded molecule inclusion. But that means if particular enzyme is absent, so particular substance cannot be degraded. In suppose like glucosidase is absent, so carbohydrate cannot be degraded. So it will start accumulating in the lysosome. So it causes lysosomal storage, right? Similarly, gout. So the urate crystals are phagocytosed and cause physical damage of lysosomes and releasing enzymes producing inflammation and arthritis. So gouty arthritis. And cathepsins are implicated in tumor metastasis. They degrade the basal lamina of the cells. Silicosis, it is also one of lysosomal enzyme disorder. Okay. And why? Because there is stimulating the fibroblast proliferation. Okay. This fibroblast proliferation causing silicosis. Actually, this silicosis related to one of the lysosomal storage disorder.